Hello everyone, today we will discuss about chylomicron metabolism. Chylomicrons are lipoproteins that are synthesized in the intestinal mucosal cells. Intestinal mucosal cells. Its main function is to transport dietary triacyl glycerol from intestine to peripheral tissues. That is the main function of chylomicrons. That is to transport dietary triacyl glycerol from intestine to peripheral tissues. Chylomicrons contain higher amount of triacyl glycerol. In addition to triacyl glycerol, these also contain cholesterol, then phospholipids, ApoB48, ApoA and ApoC2. So let's see the steps in the metabolism. These chylomicrons are synthesized in the intestinal mucosal cells. So in the intestinal mucosal cells, lipids combine with the various proteins and this forms lipoprotein complexes. Lipids combines with various proteins and forms lipoprotein complexes. These are known as nascent chylomicrons. Nascent chylomicrons. Nascent means newly secreted. Newly secreted chylomicrons. So in this small intestine, from this small intestine, chylomicrons are synthesized. So in the intestinal mucosal cells, lipids combine with various proteins and forms, this is intestine, and they forms this lipoprotein complexes. These are known as nascent chylomicrons. That nascent means newly secreted. That is, it contains triacyl glycerol, cholesterol, phospholipids, then ApoA1 and ApoB48. B48, that is known as nascent chylomicrons. Newly secreted chylomicrons. Then the polysomes on rough endoplasmic reticulum. Polysomes on rough endoplasmic reticulum of intestinal cells synthesizes this ApoB48. ApoB48 is synthesized from polysomes on rough endoplasmic reticulum. Then triacyl glycerols are synthesized in smooth endoplasmic reticulum. Triacyl glycerol in smooth endoplasmic reticulum. So in smooth endoplasmic reticulum, the lipids are incorporated with ApoB48. Lipids are incorporated with ApoB48. On polysomes on rough endoplasmic reticulum of intestinal cells synthesizes this ApoB48. Then this triacyl glycerol is synthesized in smooth endoplasmic reticulum. Then this in smooth endoplasmic reticulum, the lipids are incorporated with ApoB48. These ApoB48, then these are finally glycosylated in the Golgi complex. ApoB48 are finally glycosylated in the Golgi complex, which then packs these chylomicrons into the secretory vesicles. Secretory vesicles. The ApoB proteins, the B apoproteins, these are finally glycosylated in the Golgi complex, which then packs these chylomicrons into the secretory vesicles. Then by reverse pinocytosis, chylomicrons are released into the lymphatic system. And to circulation. So this ApoB48, these are synthesized in polysomes on the rough endoplasmic reticulum. Triacyl glycerol is synthesized in the smooth endoplasmic reticulum. In the smooth endoplasmic reticulum, the lipids are incorporated with ApoB48. These ApoB48, these are finally glycosylated in the Golgi complex, which then packs these chylomicrons into the secretory vesicles. After that, by reverse pinocytosis, chylomicrons are released into the lymphatic system, then to circulation. Then once in circulation, blood carries these nascent chylomicrons to various tissues. During circulation, they acquire ApoC2 and ApoE from HDL. So, the donor of this ApoC2 and ApoE to chylomicrons is HDL. So, from HDL, ApoC2 and ApoE. HDL contains cholesterol, phospholipids, then ApoE, then C2, ApoC2, ApoA1. ApoA1. This is HDL. This is HDL. So, from HDL, ApoC2 and ApoE to chylomicrons. ApoC2 and ApoE to chylomicrons. So, it will become mature chylomicrons. So, triacyl glycerol, C, that is cholesterol, then phospholipids, then ApoE, then C2, ApoC2, then A1 and ApoB48, and it will become chylomicron or mature chylomicron. At this stage, ApoC2 activates lipoprotein lipase. This ApoC2 and ApoE from HDL. At this stage, this ApoC2 activates lipoprotein lipase enzyme. Lipoprotein lipase enzyme. This lipoprotein lipase hydrolyzes triacyl glycerol of this circulating chylomicron to diacyl glycerol and monoacyl glycerol, and then release free fatty acid and glycerol. It releases free fatty acid and glycerol to peripheral tissues. So, ApoC2 activates lipoprotein lipase. Lipoprotein lipase hydrolyzes this triacyl glycerol to diacyl and monoacyl glycerol, and then release free fatty acid and glycerol to peripheral tissues. This hydrolysis occurs when chylomicrons are attached to the lipoprotein lipase on the endothelium. Hydrolysis occurs when these chylomicrons are attached to the lipoprotein lipase on the endothelium. This lipoprotein lipase is activated by ApoC2 and ApoC2 and phospholipids. These are activated by ApoC2 and phospholipid. These are inactivated by ApoC3 and ApoA2. Lipoprotein lipase is activated by ApoC2 and phospholipid. It is inactivated by ApoC3 and ApoA2. Then most of the free fatty acid, these are most of the released free fatty acids are taken up by the tissue cells. Smaller amounts circulate in plasma as free fatty acid albumin complex. Most of the released free fatty acids are taken up by the tissue cells. Smaller amounts are circulating in the plasma as free fatty acid albumin complex. As this lipoprotein lipase hydrolytically removes this triacyl glycerol from circulating chylomicrons, triglyceride content decreases. and the amounts of this cholesterol ester and cholesterol will double. Lipoprotein lipase hydrolytically removes this triacyl glycerol. So, triacyl glycerol content will decrease and cholesterol ester content will double. Then simultaneously, ApoA1 and ApoC2 are transferred from chylomicrons back to HDL, circulating HDL. ApoA1 and ApoC2, ApoA1 and C2, these are transferred back to circulating HDL together with cholesterol and phospholipids. These are transferred back to HDL. So, at this time, these chylomicrons are converted to chylomicron remnants. Chylomicron remnants. In circulation. These chylomicron remnants will take up cholesterol ester from HDL. From HDL, cholesterol ester is taken up by these chylomicrons in exchange of triacyl glycerol. From HDL, cholesterol esters are taken up by these chylomicrons in exchange of triacyl glycerol. Then this chylomicron remnants, it contains cholesterol esters, cholesterol, phospholipid, triacyl glycerol, then ApoE and ApoB48. This is chylomicron remnants. Then in liver, there are receptors known as remnant receptors. Remnant receptors or ApoE receptors. ApoE receptors. These receptors on hepatic cell membranes identify the ApoE of these chylomicron remnants and internalized them by pinocytosis. So, it will enter into the hepatic cells. Here it is remnant receptors. Remnant receptors. These identify the ApoE and these are internalized by pinocytosis. So, it will become CE, cholesterol ester, cholesterol, phospholipids, triacyl glycerol and triacyl glycerol. Then hepatic lipase enzyme. Hepatic lipase enzyme hydrolyzes this triacyl glycerol and cholesterol ester. Hepatic lipase enzyme hydrolyzes triacyl glycerol and cholesterol esters into free fatty acid or fatty acid and cholesterol. Fatty acid, cholesterol and glycerol. Hepatic lipase hydrolyzes cholesterol esters and triacyl glycerol. Triacyl glycerol to fatty acid, then cholesterol esters to cholesterol, then glycerol by this hepatic lipase. And these are further metabolized by liver cells. Then some of the chylomicron remnants in the liver incorporated into the phospholipids, and these are secreted from the liver as a phospholipid rich remnants or phospholipid rich lipoprotein particles. Phospholipid rich lipoprotein particles, these are having density less than 1.006. These are called as remnants. Remnants. Then this cholesterol then move back to HDL. So, this is about the metabolism of chylomicrons. Next, we will see the regulation of lipoprotein lipase activity. Regulation of lipoprotein lipase activity. Lipoprotein lipase activity decreases in adipocytes on starvation. In adipocytes and rises after feeding. After feeding, the lipoprotein lipase activity rises. So, starvation reduces and feeding enhances the uptake and storage of fat by adipose tissue. Then, but starvation enhances this lipoprotein lipase activity in cardiac and striated muscles. In cardiac and striated muscles, and enabling them to take up and oxidize more fatty acid. Then insulin also increases the synthesis of this lipoprotein lipase in the adipose tissue. So, this is about chylomicron metabolism. This is today's topic. Thank you for watching.